So the last speaker tonight is Aric Daul from the University of Minnesota, and he will tell us about the Caenorhabditis Genetics Center.
Thank you, Esther. Thanks also to Kevin and Monica for giving me the opportunity to tell you about the CGC tonight. The Caenorhabditis Genetics Center, or CGC, is the lone general stock center for C. elegans and related nematodes. We're housed at the University of Minnesota in Minneapolis. I'm the head curator, Ann Rugby is the director. Teresa, Kemi, and Julie are the technicians in the lab that prepare all the orders, prepare all the strains that get sent out to you whenever you request things. So if you've received any strains from us, it was likely prepared by one of these three. Our goals are to collect and maintain important nematode strains for the community and distribute these strains to researchers around the world. We're also working to enhance the collection of genetic tools available to the community, and we're presently doing that by using CRISPR-Cas to label existing balancer chromosomes with fluorescent markers. And as these strains are made, they are being made available through our online catalog. Our collection currently contains more than 19,000 different strains. The collection continues to grow steadily, and these strains are in demand. 92% of the collection has been requested at least once. More than 3/4 of the collection, so about 15,000 different strains, have been sent out in the last 4 years. So how do you receive the strains? How do you find what you need in this collection of 19,000 different strains? Well, there are really two ways to go about it. The first is to go to Wormbase, and you can search either by the gene, the allele, or even the strain name in their search box. And if you scroll down to the genetics section, you can see the results either in this nice graphical format here showing what we have, as well as other strains that are not currently in the collection, or in this list format down below. Alternatively, you can go to our site and search our catalog directly. Again, you may search by the strain, the allele, and when our new site comes online, there will be enhanced search functions as well. So if you're going to place an order, all orders must be placed through our website. I realize most of you know this already, but if there are any non-wormies that wandered across the hall, just wanted to give them a little info about how they may obtain strains. There's a $25 annual fee for each lab. This is basically a 1-year subscription for our services. Strains are $7 each for academic or nonprofit groups. For commercial labs, the fee is $100 per strain, and there are some restrictions on some strains that may not be sent out to commercial labs. When placing your order, you may pay at the time with a credit card. We actually prefer that because it really streamlines our handling of the invoice. But of course, you may use a purchase order and we'll send you an invoice that may be paid. All orders require a lab code. If you do not have a lab code, contact us by email and we'd be happy to help you set up your account. So, and I know the most important question from your standpoint is, how soon will you get your worms? At any given time, we have 3,000 to 4,000 worms out and actively growing in the lab. So if you order something that's actually out, turnaround's pretty quick. You can sometimes get it out within a day or two. But typically orders take about 1 to 2 weeks to prepare. Some strains will of course take longer, strains that are temperature sensitive and need to be raised at low temperature, strains with reduced brood sizes, heterozygous strains, unstable arrays, etc. Once the strains are ready to go out, shipping times do vary. If you're within the US, you'll probably receive your strains in about a week. For international labs, we allow up to 4 weeks. This is primarily due to delays in customs. Once they leave the US, we really have no control over what happens to the package. Oftentimes they don't take that long, but in some cases it does take 3 or 4 weeks for people to receive their strains. And of course we'll use a courier, FedEx, or DHL upon request, but you do need to provide an account number for us. We can't use our account and bill you for the costs. So now if on the other hand you have something you'd like to send to us, we are always looking for new useful strains that will help the community. So please donate strains if you have mutant alleles of a gene that's not yet represented in our collection, if you have a null allele of a gene that we only have hypomorphic alleles of, if you have unusual alleles, gain of function, conditional alleles, or now with the advent of CRISPR-Cas, tagged endogenous loci that would be particularly useful to the community. Hard to make mutant combinations, or new useful genetic or molecular tools, so new balancers, new deficiencies, mapping strains, etc. But of course, other strains will also be considered. Unfortunately, we cannot take everything, however. So please try to avoid redundancy, primarily with multiple lines of effectively the same transgene. Collections of similar alleles with similar phenotypes, unstable lines that just cannot be propagated long-term. I mean, we need to maintain these strains as part of the collection indefinitely. Highly specialized growth conditions, and we cannot accept entire lab collections. So we're starting to see some of the early generation of worm researchers that are hitting retirement age. Labs are closing down due to loss of funding, et cetera. But we simply can't take on hundreds of strains at once. But if you have something you would like to donate and you're unsure about your strains, just ask us. If you do have something you would like to send to us, please submit a completed strain donation form before sending the strains. Please don't send them without prior approval. The donation form is currently available for download from our website, and our new site will use an online submission form. So you'll be able to just go into the menu, click it, and it's going to be an online fillable form that will get submitted to us, and we'll get back to you. No strain will be accepted without a complete genotype. This is really critical. Your strains are only as useful as the information you provide to the rest of the community. Genotypes need to conform to established nomenclature, and also be sure to include any co-injection markers or transformation markers used if you are sending us transgenic lines. If you have any questions about nomenclature, Wormbase has a lot of information that's kept pretty up to date. You can find a link to their nomenclature page in the bottom left of nearly any page on Wormbase. Large collections will need a prior approval by the CGC, and we may consult the advisory board, and you will be notified if your strains are accepted or declined. You're probably wondering what happens to all those worms when you send them to us. When I receive a new strain, the strain is examined, cleaned if necessary, and frozen. We store multiple aliquots both in a -80 and in liquid nitrogen tanks within our lab. Additional aliquots are stored in huge, large capacity liquid nitrogen tanks in secondary sites on campus. These are in different buildings elsewhere on campus. And to prevent against catastrophic loss at our location, we also send a single aliquot to the USDA germplasm collection in Colorado for safekeeping. So the entire collection is backed up and preserved in the USDA as well if something really catastrophic happened to us. Although I suppose I wouldn't be worried about it anymore if that happened. So, of course, we don't want to just keep these frozen away. The whole point of this is to collect strains and make them available to you, and you have been keeping us quite busy. So once again this year, we're up around 30,000 strains. In fact, over the last 4 years, we're averaging 30,000 strains going out each year on about 7,500 orders each year. This comes out to an average of about 120 strains shipped out per day. And while I find that to be an impressive number, it's nowhere near our all-time single-day record of 619 strains that were shipped. Among the most popular strains going out, a lot of these strains are models for aging studies or human disease. N2 is still a king. And not surprisingly, a lot of these strains are also among the top 10 strains of all time. It's hard to believe we sent out N2 more than 5,000 times over the years. You might be wondering, where are all these strains going? And some of the increased demand for strains is driven by an increase in the number of users. The worm community continues to grow steadily, and at the end of May of this year, despite the scale being off here, we're at just under 5,000 labs, and in the last few weeks we have actually gone over the 5,000 lab mark. So we have more than 5,000 labs registered with us. 1,200 of these labs have alphabetic designations, meaning they're actively naming strains and new alleles. And while just over half of these labs are within the US, we're clearly a global commodity. We've sent strains to 72 countries around the world. Shipping internationally is difficult, however. As any of you outside of the US probably know, clearing customs is our biggest challenge. Rules are constantly changing. Rules vary by country and sometimes even by the port of entry within a country. So if your country requires any special forms or special import permits, please send us copies of them so we can include them with your strains when we send them. Please be sure to provide a complete shipping address, including a department name, a room number, etc. Give us as much information as possible so that when these strains make it to your country, they have the best chance of getting delivered to you as quickly as possible. And if possible, please provide a FedEx or DHL number, especially if you're in a particularly hot or cold climate, such as India in summer, Canada in winter. Just do what you can to help keep those worms alive. Now, the top 10 users, when you look at this list, you also see the international nature of our customer base reflected in this list as well, with 4 out of the top 10 labs are outside of the US. Gary Ruvkin's lab has been in the top 10 every time we've presented this data since 2003. But in spite of being in the top 10 for the last 13 years, he's still not number 1. He's still well behind Henrik Bringman. So if there's anyone from the Ruvkin lab out there, you might want to tell Gary that he needs to step up his game if he's going to get to be number one. But seriously, there's clearly a lot of excellent work being done with all these strains we're sending out, because we're seeing over 1,000 worm papers each year. So these are papers that include C. elegans in the abstract when we search on PubMed. And we're happy to see that the number of papers that are acknowledging us is going up, but there's still a long way to go. Please acknowledge us in your papers. This really helps us maintain our funding. This is a key stat when we apply for renewal of our funding with the NIH. Okay, I'm going to shift gears here and tell you about the balancer project that I mentioned earlier. As we know, as any worm people know, the collection of balancers available in the worm community is lacking. Don't tell the fly people that worms aren't perfect. I've been trying to convince them of this for many years. The first aim, we're using CRISPR-Cas to label existing balancers with easy-to-detect fluorescent tags. And secondly, we've recently initiated a collaboration with the Mitani lab to use CRISPR to generate new balancers that provide better coverage along the chromosomes. So most of this work has been done by Julie Knott in the lab, who's really a driving force behind this project. And her initial efforts were using EFT3 to drive expression of TD tomato or GFP. But in a single copy, this promoter has rather low levels of expression. So we've since shifted to the MYO2 promoter, which gives us a much more robust signal. So we now have these two balancers labeled with MYO2 GFP. And using a strategy that allows us to target the regions of chromosome 3 and 5 in these other balancers, these are in progress and some of them should be available soon. And I know some of you in the audience will be happy to know that we also have SZT1 that's currently in the works and should be available soon. And next up, we're going to be labeling these with red to make red versions of these as well. So if you're working with green transgenes, you're able to detect the balancer versus your transgenes. And just one other thing I wanted to mention quickly. Some of you may have already encountered this, but the NIH has initiated a new set of publication guidelines. This has been adapted by some journals, and it's basically a unique identifier for key reagents, including worm strains. This is known as the Research Resource Identification Initiative, or RRID. We find this to be a bit redundant with the nomenclature system already in place in worms, where we have unique identifiers for strains and alleles and everything else, but we've agreed to go along with the NIH guidelines. So RRID information for the worm community is being managed by Wormbase. And as such, the RRID tag for the worm community is going to be RRID:WB for Wormbase, followed by the normal strain name. So for example, if you were publishing RG1228 somewhere in your paper, if the journal requires it, you'd have to provide the RRID tag WB RG1228. More information about this can be found at scicrunch.org. So with that, I'd like to thank the rest of the CGC staff, many people for database support, Mark Edgley, Tim Shadle, and a lot of people at Wormbase, and particularly Mary Ann Tooley, the advisory board, in particular Ivy Greenwald, the chair. And if it were not for the continued financial support of the NIH, we wouldn't be here tonight. So with that, take any questions if you have them, but otherwise, thanks to all of you. We are a community resource, and it's the strains that you contribute to us that really let us do what we do. So thank you. There was a stretch where some places, like I think some people at the NIH were requesting that we mark do not x-ray. There are a couple of people that request us to mark do not x-ray. I'm not aware of it being a common problem, but if it's a concern, if you make a comment in your order, we'll label the envelope. We can't promise they're not going to do something bad to it when it arrives. But if you have special instructions like that, let us know and we'll do what we can to accommodate them.
So I just want to applaud you and the CGC for doing a great job. I think it's really important for the community. I also just want to mention the policy that we use in our lab for sending strains to you, which is that whenever we get a request from anybody anywhere in the world, and if it's not at the CGC, then we make 2 copies. We send one to the person who requested it and one to you. And that means that anything that's actually being requested goes to CGC. And I think that this is a lot easier than when I retire, sending you all the strains that I think somebody might want. So people might want to use that kind of guideline as well for, and you've always accepted the strains we've sent at least.
Yeah, so again, I think that's a pretty good measure. If there's demand from within the community, especially if it's something you're getting multiple requests for, let us know. Basically, if someone can make a compelling argument why we should have it, even if it's, a second hypomorphic allele or another transgene that's very similar, if there's a compelling argument for it, we'll probably take it. But again, it's just the blanket approval of taking, a whole collection of the same transgenes or 3 lines of the same thing that space is a concern. I mean, right now we have 4-80 freezers. We've got 8 what we consider the smaller nitrogen tanks, and again, 2 of the huge tanks that weigh as much as an SUV, so we're really restricted on where we can expand. We're not in danger of running out of space immediately, but it is something down the road, especially with the advent of CRISPR-Cas, and generating new strains is probably going to explode. So. All right, thanks.
Then thanks everybody, and thanks to speakers for this very exciting session, and see you all tomorrow. Good night.